I am now going to pass you over to Judith Worthington. Judith, I'm going to stop sharing and then you're going to share your screen.
Okay, let's hope it works.
Okay, good to go? Bear with.
Okay, is that showing okay, the slides? Right, good evening, everybody. Thank you for joining us.
Judith, we're not seeing your slides just yet.
Oh dear, that's not good, is it?
Don't worry, it'll come together, I'm sure.
Let's try doing that again.
Susan is Judith's co-host.
It worked perfectly when we did the rehearsal, didn't it?
It did work so well, just at 6:10.
Is that working? No.
No, so okay, bear with us, everybody.
Oh, hold on.
That's it.
Hey, there's another button to click. Oh dear. This is filling you all with confidence about me, isn't it?
No, that's fine. IT's not your speciality. You don't claim that. And just a final thing to move on to your view for your slideshow.
Okay.
Fantastic. Thanks,
Hey.
Judith.
Okay, thank you, everybody. Thank you for the invitation to talk to you tonight. I hope you find this useful. I am based in Manchester at the Royal Infirmary, but this is a generic talk that would apply to anybody wherever you are in the UK. And there will be my equivalent working in a lab near you as well. If you want to ask, more specific stuff about yourself. So the topics that I'm going to cover this evening, I'm going to start off and I'm going to do the science bit. So I'm going to tell you about tissue types, HLA types, how we match, what are antibodies and why they're important. And then I'm going to talk about cross-matching. And then once I've sort of set the scene with the science bit, I'm going to talk about the biological factors that will affect how long you might wait for a transplant and the effect that those have. So first of all, what is a HLA or a tissue type? So a HLA or a tissue type can be thought of as like a barcode on products in the supermarket. So this barcode is present on all the cells in your body. So it's expressed on the cells in the kidney as well. And it's absolutely unique to every individual unless you have an identical twin. So tissue typing is a series of DNA-based tests that identify this barcode or work out what someone's tissue type is. And because we use DNA to do these tests, we would normally use a blood sample to carry out the test. But we can also use buccal swabs or we can use blood spots. And we've actually even validated the test to be able to tissue type somebody from their toenails, which we have used on occasion when people have received transplants overseas. Before anybody goes on the transplant list, we always do their tissue type twice. We do it on 2 separate sets of bloods. And that's because we want to be absolutely sure that we've got absolutely the right tissue type for the patient. So we want to make sure there's been no sample mixes up either in the clinic or in the laboratory. So we always do type everyone twice. So then what happens is your immune system acts as like the scanner. So it scans the barcode and makes sure that the barcode that it detects is the one that identifies you as you. And if it detects a barcode that isn't yours, it will get the immune system going to remove any cell that doesn't have that barcode. So anything that's different from your own. So what does a tissue type look like? Well, this is a tissue type. This is my tissue type, actually. I don't feel like I'm sharing anything particularly confidential with you because, as you can see, it's just like a series of numbers, letters, colons, asterisks. So it doesn't really mean anything to anybody unless you're an H&I specialist, and then you might know which ones are more common than others and which ones are rarer than others. or you might even be able to identify somebody's ethnic background from the tissue type, because certain tissue types are more common in different ethnic populations. So, as I say, probably doesn't mean an awful lot to anybody, but that's what a tissue type looks like. And there are lots of genes that make up your tissue type that are given different numbers, so A, B, C, D, P, alpha, sorry, letters, not numbers. But in terms of transplantation, we really just concentrate on 3 main genes, which is HLA-A, HLA-B and HLA-DR, and they're actually referred to as the classical transplantation genes. So where does your tissue type come from? It's determined genetically. You get half your tissue type from mum and half your tissue type from dad. And the 3 genes are inherited together in strings or haplotypes. So if we look at our family tree that we've got here, we can see somebody's Homer's tissue type here, Marge's here. And then Homer could donate either the red string and Marge the blue, or Homer could donate the yellow string and Marge the orange. So in any mum-dad combination, there are only 4 possibilities that the children can be. So in this example, we can see that Lisa has inherited the red from Homer and the orange from Marge, as has Bart. But Maggie's inherited different haplotypes. So she's inherited this yellow one off Homer and the blue one off Marge. So you've got a 1 in 4 chance of being exactly the same tissue type as your brother or sister. You've also got a 1 in 4 chance of having absolutely nothing in common with them whatsoever. And it's just that you've inherited different things off your mum and dad than your brother or your sister. Or you can be half the same as your brother or sister. If you had extended family, so I know there's been some questions about cousins, the possibility still exists that you could all have inherited the same haplotype. So Marge's sisters, if there's any Simpson aficionados out there, I can't quite remember names, I think it's Patty and Selma, may have inherited the same tissue type as Marge, or they may be 1 in 4 chance of being the same. So that's where your tissue type comes from and how you inherit it. And then we talk an awful lot about matching for transplantation. It's even in the title of our talk tonight, but actually what's important is mismatching. So if Bart needed a kidney transplant, this is Bart's HLA type. So he has 2 A's, 2 B's and 2 DR's because he's got 1 of each off mum and 1 of each off dad. And then we have dad here. So what's important is what dad's got that Bart hasn't, because that's what Bart's immune system would recognise as being not of Bart's barcode and recognise as foreign, and then get the immune system to destroy that foreign HLA type. So here we've got the A2 in common with both, but that doesn't really matter. What matters is what Homer's got that Bart hasn't, because that's what Bart will raise the immune response to. So A3 is a mismatch. Here we've got 15 and 44. It's the 15 that is a mismatch. And here we've got 4 and 7. It's the 7 that's the mismatch. So this is then abbreviated into a 3-number code. And we can go through and work out the 3-number codes for each of them. So we said there was 1 mismatch at A, 1 mismatch at B, and 1 mismatch at DR. That gives us a 3-number code of 111. Whereas Lisa has no mismatches with Bart because they're exactly the same. So there's no mismatch at A, no mismatch at B, and no mismatch at DR. So it's a 0, 0, 0. And that's
Judith,
yeah.
Sorry, can I just jump in? Does everybody know who Bart and all the names of people are? I wonder if you could use your, can you use your pointer?
I am doing, is it not showing on the?
No, because I must admit, I'm thinking who's who.
Oh, right, sorry. No, that's fine. Thought everybody knew who the Simpsons was. So if we say our skateboard dude at the top, I'm trying not to be rude about some of the others. So we've got skateboard dude who needs a kidney, and his dad is offering to donate him a kidney, and then we can work out the 3 number codes. So we can see that Homer has A3, dad has A3 that dude hasn't. Dad has B15 that dude hasn't, and dad has DR7 that dude hasn't. So he has 1 mismatch at A, 1 at B, and 1 at DR. If we move on to the sister with the pill necklace, then you can see that she's exactly the same tissue type as our skater dude. So that is a 0, 0, 0. So no mismatch at A, no mismatch at B, and no mismatch at DR. And baby cis is completely different than our patient. So there are 2 mismatches at A, 2 mismatches at B, and 2 mismatches at DR. Is that okay? So because you've got this, then you can say that there's a total of 3 mismatches with dad, no mismatches with the sister, and 6 mismatches with baby cis. And the reason that we do look at how well matched people are is because it does have an influence on how well the graph might do going forward. So this is data from a study that's based in Germany, but they collect data from all over the world. So they have huge, enormous data sets. So they're really useful for showing the effects of matching on graft outcome. So here there are 6 lines on this graph. The top line, the black line, being where there's no mismatch. So the bit where like sister to skater dude, or the sort of mucky green colour where there are 6 mismatches at the bottom of the graph, which is baby cis and skater dude. So at the beginning of the post-transplant journey, this doesn't really have a huge effect. The lines are quite close together, but you can see by the time you get to 5 years post-transplant, the graphs that do better are those that are 0 mismatch, and the graphs that do not as well as that, but still very good graft survival at 5 years, are the ones that are mismatched. So that's why we do take into consideration matching. So that's what a tissue type is, and where it comes from and why it's important in transplantation. The next thing that we need to think about is what are HLA-specific antibodies. So these antibodies are found in your blood. They're what the immune system uses to destroy the foreign barcodes in your body. So the antibodies bind to the cells that the immune system has recognized as not having your barcode, and then they will destroy those cells. So they're absolutely crucial for fighting viruses and infections, because otherwise you would succumb to every virus and infection that there is. But unfortunately, they're also very good at recognizing transplanted organs as not being as being foreign. So they also attack the transplanted organs. So the sample that we need from patients to screen for HLA-specific antibodies is a clotted blood sample. And that's just a tube with no anticoagulant in at all. How do you get HLA-specific antibodies? Well, these can only occur when your immune system sees somebody else's barcode or somebody else's HLA type. And there's only 3 mechanisms that can happen through. So the first one is pregnancy, because when mum's pregnant, the baby is half mum, half dad. So the mum's immune system will see the dad part of baby and may produce antibodies. These antibodies are not harmful to the baby in any way. They don't cross the placenta. They're just something that we have to bear in mind if you need a kidney transplant. The other way, which is much less common nowadays than it used to be, is blood transfusion. That's because normally blood transfusions are given using a white cell filter, so the impact is lower, but you can still produce antibodies if you have a blood transfusion. And the third way is a transplant, because you're almost certainly going to be mismatched in some way if you receive a transplant, unless you've got that HLA identical twin that you can have a kidney from. And the more pregnancies, transfusions and transplants that a patient has, the more likely they are to produce antibodies or produce more antibodies. So you may know that we ask for samples on a regular basis from patients whilst they're on the transplant list. We do this, and you may say, well, I've not had any pregnancies, transplants or transfusions. But the reason we do this is the amount of antibody can change over time. So our tests are only actually showing just a small snapshot of what's happening at the time that sample's collected. And whilst you might not have had a pregnancy, a transplant or a transfusion, you may have had some other infection, whether it's related to peritonitis or just a cold or another viral type of infection, and that may cause your immune system to wake up again and start producing antibodies again. So the tests that we use to look for antibodies give us a numerical reading, which is these numbers that are on the graph here. And the test gives us you could track the test, and it will follow this orange line. And the blue line is the cutoff for the test. So below the blue line is considered to be negative, and above is considered to be positive. And then over the years, you can see that this particular antibody profile has gone up from 2015, peaked at 2018. So this is when the antibodies are at its strongest. And then over the next few years, it's gone away again, fallen below our cutoff, and then it's popped back up again in 2021. And as I say, these won't be direct exposures to another person's HLA. They may just be because of responses to non-specific infections. So that's why it's really important that we get regular samples of patients while they're on the list, so that we always know exactly what their antibody profile is. And the final thing that we always do is the immediate pre-transplant test. So once an offer comes in for a patient, there are some tests that we may still have to do on the day. We may need to do a screening test, so we may need to check what your antibodies are currently. That takes us about 2 hours to complete that test. It's a very quick test and gives us a final go ahead or not. In some cases, we might need to carry out a cross-match test, and this takes a little bit longer. That takes about 4 to 5 hours to complete. A cross-match is, well, when I started work here, it was described to me as like doing a transplant in a test tube. You take cells from the donor and mix them with the sera that we have in the freezer that we've collected over the years from the patients. And then if your sera attacks the cells and kills them, that's what would happen if we put the kidney in. So this is why it's been described to me as like a transplant in a test tube. So if your sera contains antibodies that are specific for that particular donor cell, then they will kill the cells, and down the microscope they look this red colour, red-orange colour that is in the top slide. And if you don't have any antibodies and it doesn't attack the cells, the donor cells, then the cells stay alive, and down the microscope they're this green colour. So the red is a positive cross-match, means the transplant can't go ahead. Green is a negative cross-match, means the transplant can go ahead. And that's sort of the immediate pre-transplant test that we do for everybody, whether virtually by looking at your antibody profile or by actually doing the wet cross-match test. So that's sort of the end of the first part of the talk, giving you the background and all the science and everything. I hope it's been clear, but hope we'll come to questions and it'll be obvious if it hasn't been. Having a cursor, a pointer might have helped, but
Judith, it was truly fascinating. I myself am a kidney donor, and even throughout that whole process, I never learned as much as I just have tonight. There was little bits that no matter what, you just couldn't understand. And certainly having that family with numbers started me to understand what a 0-1-0 match is and things. Or should it be mismatch? I'll have to rewatch. But certainly there was so much information. It was fabulous. There is some questions starting to come in. And again, there's still people joining that are maybe later home from work. So we'll start just in the order they've come in. And
Shall
Kay's
I
asking,
stop sharing?
Kay was asking there.
Sorry, are you, sorry, jump in. Are you resharing after? Are you sharing the second part afterwards, Judith?
If the questions, if you don't think the questions are going to be answered by the second part of the talk,
Yeah,
or
okay.
I can carry on.
No. I think it's a good. Yeah.
There were actually asking about the antibodies, and it was more when you have antibodies, are they there forever? Because you did just show in that chart where they rose and they come down, and I think on that, would it give someone hope that that can help them?
Yeah, unfortunately, there's no one answer to this. Some people can have 3 pregnancies, 2 transplants, multiple transfusions, and never produce an antibody. And then somebody else can walk past the blood bank and just sniff the air and they produce antibodies. It's probably the same way in that some people have responded differently to the COVID vaccinations than others. Some people respond strongly, some people don't. And then some people's antibodies persist for a long time, and some people's will move, as I showed in that graph there. But there's no easy way of knowing who that's going to be. It's all about how strongly your immune system might respond and how long. And also it makes a difference about how the sensitization happened. So a transfusion related sensitization is more likely to go away again, whereas a pregnancy or a transplant transfusion is more likely to persist.
Lovely. And also changes can happen to that potential donor. Kim's aware that her husband is a potential match, 101, but he's recently had a partial cornea graft. So it was transplanted materials, but it doesn't need any anti-rejection drugs, etc. Could that potentially affect his viability about being a donor?
So sorry, is the person who's received the cornea transplant the donor or the recipient?
Donor.
The donor, right. So that won't affect the potential donation in any way. If it had been the other way around, if it had been the recipient who'd received the cornea graft, then potentially, because that's a source of sensitization. But as the donor receiving one, that won't influence them from a H&I point of view, no, an immunological point of view.
And just relating to those kind of final slides that you did share with us about mild positive reactions during a cross match, does this mean that that donation wouldn't go ahead if that was the case, if there was what they're describing as a mild positive reaction during a cross match?
So the cross-match results I showed you there are a cross-match called a complement-dependent cytotoxicity cross-match, and that's like our gold standard. If you have a positive cytotoxic cross-match, then that would be extremely high immunological risk. There'd be a very high chance that that graft would reject, even to the point of hyperacute rejection. So, but there are other cross-match tests that are not as sensitive, called flow cytometry cross-match tests, that pick up weak reactions, which can be managed with the immunosuppression that is given to a patient post-transplant, or can be managed if you're aware of them pre-transplant and you have the time, by actually performing plasma exchange, which would remove some of those antibodies and get them to a lower level so that the transplant would be safe to proceed, and then managed post-transplant with the immunosuppression.
So that that could be a scenario. That's really a question for the individual's transplant team, where they have those results sitting in front of them.
Yeah.
And an interesting one, Sarah's asked in the chat, can warfarin affect an antibody test? So can warfarin or any other medications that we may take affect that test?
No, it shouldn't. We choose to test serum, so that's blood that's clotted, and then take the residual off and test. But if that blood hadn't clotted, the residual would be called plasma, and you actually get the same result with the test. It's the same, whether it's plasma or serum.
That is superb. There's lots more questions coming in there. there are relating things that I think you will be covering. So I think we'll move on and come back to those questions. Thanks,
Okay.
Judith.
So are the slides back?
Yes.
Hooray! First time. So I'm just going to define a couple of terms that you may hear talked about, because in order to go on to the next bit to talk about what affects how long you might wait, you sort of need to know what these are. So there's one called calculated reaction frequency, which also historically used to be called panel reactive antibody. So this looks at how many antibodies you have and how many of the last 10,000 UK donors you would have been antibody positive to. And it comes out with a numerical output, which is your calculated reaction frequency, or CRF. So if you would have antibodies that would react with 10% of the last 10,000 donors, your CRF would be 10%. If you have an antibody that would react with 50% of the last 10,000 donors, it's 50%. And this pie chart just shows the distribution of people who are currently on the transplant list in Manchester. So you can see the majority of the transplant list is made up of patients who have antibodies but are classed as, you know, some antibodies but not lots of antibodies, so 0 to 84%. And then we have 3 smaller cohorts who are our highly sensitized patients, where their CRFs are 85% or greater. Just another way of looking at that is this top one where all the boxes are green is because this is all the different HLA types in the box, and they're all green because the patient has no antibodies to HLA-A at all, so they have 0% antibodies. Whereas in the second box we can see that quite a lot of the boxes are marked as red, and that's because they have antibodies directed against those HLA antigens, so they wouldn't be acceptable in a donor for them. So that's like 90% of the HLA types are unacceptable, so that's 90% CRF. The other term is matchability scores or matchability points. So this is a score between 1 and 10, and it reflects how difficult it would be to find a well-matched donor for you. So it takes how long people have waited and divides them into 10 epochs or 10 sections. So the first section is the 10% of the waiting list patients who matched with the most of the 10,000 donors. And the other end of the scale, we have the 10% of patients who matched with the least number of the last 10,000 donors. So if you're in the bottom 3 sections, 1, 2, or 3, you're classed as easy to match because you would have matched with the most of the 10,000 donors. 4, 5, 6, 7 is called moderate to match, and 8, 9, 10 is called difficult to match. So just gone through those terms because I'm just going to talk about biological factors that affect how long you can wait. And the 3 main ones really is what your blood group is, what your tissue type is, and what antibodies you've got, how many antibodies you've got, and what they're directed against. So ODT, which is the body that holds all of the transplant waiting list in the UK, have on their website something called a matchability calculator. And this calculator is basically an Excel spreadsheet, and into it you can put the ABO, the HLA type, and any HLA specific antibodies. So the 3 things that are circled in red. And then the Excel spreadsheet will then compare that information with the last 10,000 UK donors and come out with how many the patient has antibodies against. So what is their CRF? Are they easy, moderate, or difficult to match? And then it actually comes out with a numerical output of how many donors would have been suitable for that recipient. So here we've got somebody who's blood group O, we've got their tissue type in, we've got their antibody profile registered. They're highly sensitized, they've got a CRF of 90%, they're classed as moderate to match. And of the last 10,000 donors, 484 would have been matched. So this calculator is useful because it's individual. It's looking at the actual factors that are going to affect each patient. It's easy to use. So if we talk about blood group first, as you know, some blood groups are more common than others. And so not all blood groups are compatible. So you have a blood group donor who's O is called the universal donor, would be compatible with a recipient of any blood group. And conversely, if you've got a recipient who's blood group AB, that's known as the universal recipient. So they're compatible with any blood group donor. But you need to be ABO compatible before a transplant can go ahead. And because some are more common than others or more frequent than others, it will affect how long you might wait. So the blood groups that have the shortest waiting time statistically is blood group AB because they are the universal recipient. And the blood group that has the longest waiting time based on data is the recipients who are blood group B. So if we use that calculator just to show what sort of effect that can have. So on the left-hand side of the screen, we've got recipient information that we've put into the calculator for somebody who is blood group O, put their tissue type in, put their unacceptable antigens in, and then it's saying that just over 2,000 of the last 10,000 donors were suitably matched. If we leave absolutely everything else exactly the same, but change that blood group to B, you can see that the number of potential donors drops quite significantly, and that's why blood group Bs tend to wait longer for a graft than blood group Os. So that's just demonstrating the effect that ABO can have on how likely you are to get a match. So the other thing, other factor that we said that might affect how long you might wait is how many HLA specific antibodies you have. So if you think back to that pie chart, so where the majority of patients fall in this 0 to 84% calculated reaction frequency, the median wait time for these patients, again, this is national data, is about 2.5 years. Whereas the patients who have 100% CRF, their median wait time is about 6.5 years. So again, we can show this in the calculator. So this is the same patient, but we're giving them different antibody profiles. So on the left-hand side of the screen, we've got patient with no antibodies, just under 5,000 potential donors in total out of the 10,000. Then in the middle, we're showing that if we use keep everything the same, but just add in some more antibodies to that patient and up their CRF to 50%, the number nearly halves. And if we keep everything the same again, but add some more antibodies in, so they've had some more transfusions or they've had a transplant and the CRF has gone up to 93%, then we can see that the number falls again of the number of donors that would be suitable. And the final thing that affects how long you might wait is how common or how rare your HLA type might be. Because some are more common than others. So it's the same as blood groups. If you think about ABO blood groups, you know that O is the most common blood group, AB is the rarest. It's the same for HLA types. HLA-A2 is one of the most common HLA types. Almost 50% of the Caucasian population are HLA-A2, whereas you can see from this bar chart, only about 25% of the population are HLA-A3, and maybe only a very small proportion of the population are HLA-A80. So again, we can demonstrate this in the calculator. So we've put the patient information in, and the only thing that we change this time, we leave their blood group the same, we leave their antibodies the same, but we change their HLA type to some common ones, not so common, and some rare ones. So when we put in the more common HLA types, they're classed as easy to match by the calculator. And this is the number of donors that would suitably match for them. If we change the HLA type slightly to include some slightly rarer antigens, they become moderate to match. Now you can see that the total number of patients or donors remains the same. What changes here is not the total number, but how well matched they might be. So a level 1 match is that 0, 0, 0. So you can see that when the patient is classed as easy to match, there's just under 100 of patients would have been a 0, 0, 0, donors would have been a 0, 0, 0. When we change them to moderate, none of them were that perfect match, and we have less of the next level of matching as well. It goes from 248 to 75. And when we put in the very rare HLA types, again, the total number of donors doesn't change. It's still that 2,500. But again, it's getting harder to find the better matches because we're including the rarer HLA types.
Judith, can I just jump in here? Because somebody's asked this question, and I must admit, I was thinking it myself. Who has access to this Excel spreadsheet, you know, sort of / calculator? And can the public use it?
Yeah, it's on the internet. It's actually on what's called the ODT clinical site, but you can still access that. I use it frequently from home. It doesn't have to be through an NHS website or anything. What you do need to know though is the information
is the data. That's what I was just thinking.
Yeah. There is another tool on the ODT website as well, called Relative Chance of Transplant, which looks at people with the same or similar profiles and looks at what happened to them after 0, 1, 2, 3, 4, 5 years. Again, that's a really useful calculator, very visual, showing you how many people have been transplanted at the end of each year, how many are still waiting, how many have been removed from the list. But again, you need the information to input into it. So it's freely available to everybody, but you need to know what you're putting in.
And the information that's required to go into the calculator comes from where?
So that would be your blood group, your tissue type, and any HLA antibodies that you have. And
is this something that's readily available to people on the transplant waiting list if they wanted it? Could they obtain that?
Yeah, it's their data that, you know, no problem there.
Thanks, Judith.
So I mentioned there's an ODT clinical website. There's also this website which has lots of information on, lots of statistics, lots of reports, lots of outcome data, median wait times by centres. It's a whole wealth of information here, and a lot of the stuff that I presented will have come from here as well. So that's me done.
Absolutely fantastic, Judith. And just in the middle of all that chat while I was going through trying to pick out the questions already, you've got a thank you in there. Thank you, Judith, for clearly explaining what goes on behind the backgrounds. And I think that was the aim of tonight. There's certainly plenty of questions in amongst there, so we'll have a good look and see what it is. Lots of people are saying that calculator sounds good. Can I put the caveat in there? If we don't know how to work something and we're guessing, we could frighten ourselves. So maybe sometimes it's better left to the nurses or the doctors at Transplant Clinic. So looking at what we've got coming in that chat, we'll just... Someone's asking their daughter, and I'm sure there'll be many on the call whose children are affected, if a child needs a transplant, would mum and dad, who are all A positive blood groups and never had any blood transfusions, etc., is there a high chance there would be a match without doing any tests? to see if they'd be a suitable donor. So mum, dad, child who will need a transplant in the longer term, all A positive.
Yeah. So if there's no if the patient is HLA specific antibody negative, then there's no immunological barrier there for transplantation. And because of the way you inherit your tissue types, you will be at least a 50% match, if not better, depending if you happen to have inherited the same thing from both parents.
So it's a bit like when we think of The Simpsons, where we had Dude.
Yeah.
And Dude's dad, etc., because we didn't put Dude's wife in there. Mum wasn't in amongst that chart, but it gives us an idea of those different parts that come from different people.
Yeah.
Now, going back to that calculator, Tracy's asking the question, at what point do you get these figures? When's your profile done? Is that when you're kind of listed in the discussions about the potential of being listed for a transplant?
Yeah, so obviously I can only talk about what happens here at Manchester, but before anybody goes on a transplant list, they go to a transplant listing MDT, and that information is all there at that point. And because it's used to inform the discussions that we have as we go along through the transplant MDT. So that's something that we prepare before the MDT.
Thank you. And Pete's just popped in there. If you want to go through your times in the chat, we've all got access to that chat. It's 7:14. Pete Revel popped in a link to that calculator. He's a verified member. He's the NKF representative on the call tonight. Thanks, Pete.
Susan, people aren't letting go of the calculator, aren't they? And I think the question that is being asked is, how do they obtain the information? Where from? Who would they contact to get the information that you think is required to go into the calculator?
I would recommend they contacted their nephrologist. So the way we deal with it in Manchester is that if somebody has these specific questions, the nephrologist gets in touch with us. We then provide the information to the nephrologist, and then the nephrologist can sit down in clinic and talk the patient through it, because obviously it's a lot of information and it's quite complex. So we always find it's best if it's done at a clinic appointment.
Fantastic.
Thanks.
And any other eagle eyes there? Pete, Jade, can you see anything else that's standing out to you?
No, I think you've covered it really well, and what an informative webinar this has been, hasn't it?
Yes, certainly. Is there anyone at all who'd like to perhaps ask a question live? If they want to raise their hand. Now, Jane had said earlier how we do that. At bottom or top, you'll see a little smiley face with reaction. You can hit that button and it gives you, Michelle has raised her hand. Michelle, would you like to unmute yourself? And we'll always say, I don't have a bleep button in the house, so please do ask your question.
I've actually registered for both myself and my husband, and it's my husband Bobby here that's on the transplant list, and he has a question.
Yes, hi Bobby.
So good evening, and thanks very much for the presentation, quite informative. To the doctor and the specialist, is there any way that you can retrain the immune system so that you can hide the organ? And you also talked about the fact that an unborn child is protected because it's got a barrier called a placenta. Going forward, is it not possible to protect other organs in a similar way? No, can you not come up with a dummy placenta to protect everything?
Yeah, I mean, pregnancy is a sort of very immunologically unique situation. It's the antibodies that don't cross. So the placenta is the link between mum and baby that gives baby all the blood supply and oxygen supply and nutrient supply that they need whilst the baby's growing in the womb. But the antibodies can't cross that barrier. It's like a one-way street with the antibodies.
Well, I was just trying to think going forward, you talk about organ rejection because the person's immune system attacks the organ.
Yeah.
I'm just
No,
thinking,
I mean, there's a lot of research going on, and there's HLA incompatible transplantation, where the antibodies are temporarily lowered by plasma exchange or the use of drugs such as rituximab. And then you've sort of created a window of opportunity and the transplant goes ahead. So that is of increased immunological risk. It's not standard immunological risk, it's increased, so there's a higher chance of rejection. There are very new drugs that are not in use yet, but I know that the information is circulating around about a drug called imlifidase. Imlifidase would facilitate high immunological risk transplantation. It's not happened in the UK yet. There are some small studies, but has recently been approved by NICE. So there is research going on, and then there's research going on that has been going on for a few years now about actually growing organs which would be immunologically naive, so they wouldn't have any HLA on them, so that they wouldn't reject. But it's all very much research work at the moment.
Thank you very much.
It was a good point. I think you do raise an interesting concept. So there was a question about pregnancy, and I can't seem to find it now. I think somebody was asking somebody who's had a pregnancy, are they going to be able to get a match? I mean, you did cover it, Judith, but I think maybe you could just sort of touch upon that in terms of antibodies through pregnancy.
Yeah, I mean, pregnancy-related sensitization, not everybody will produce antibodies, and also it will depend on how many mismatches there are between mum and dad. So, you know, if dad has happened to be quite well matched, or even the baby is well matched with mum, then there is nothing for them to produce antibodies to. So it's going to be different in every situation. And pregnancy-related sensitization tends to drop below that cutoff of the test, so you don't detect it in the circulation, but it does, if you're re-exposed to it through transplantation, can come back.
What you say, it doesn't automatically prevent you from having a transplant.
No.
I mean, if the person's still in the meeting, I did have a pregnancy, and I'm 5 years post-transplant. So, and I'm sure there are other people in the meeting that have had pregnancies and had transplants.
I see we've got 3 raised hands, and I think those 3 raised hands will maybe take us to full time, 7:30. We will try and fit in some more from the chat. So I'm aware that Roger, Raymond and Gemma have raised their hands, and I'll do it in the order that I noticed the hands. So I'll come to you, Roger, if you'd like to unmute yourself and perhaps switch on your camera, or leave it off if you want to be anonymous.
Hello there. Thank you for your very lucid talk. I was wondering if you could explain why of the genes you only focus on A, B and DR and not the other types.
Okay, well, that's because the expression of the others is lower. So what I mean by that is, if you think of the cell surface and the HLA antigens on them, you get a lot of A's, a lot of B's and a lot of DRs, but you don't get as many C's and DP's and DQ's, and that's why they're not taken into account as much. The other thing as well is, if you're trying to match every single HLA locus, then you're going to be less likely to find a match. It becomes very difficult. So I'm sure you've heard about people trying to find matches for stem cell transplantation. In stem cell transplantation, they have to match 12 HLA loci, and that's why you hear about these national appeals for people. So it's partly an immunological reason, because they're not as influential in graft outcome, and partly because a logistical reason, because to try and match all 12 loci would make it difficult to find anybody a match ever.
Well, it's born out of pragmatism, basically.
Yeah, but there is immunology behind it as well.
Oh, of course, yes.
Thank you for that question, Roger. We'll move on to Raymond now. You just need to unmute yourself, Raymond. Depending on your device, there'll be a little button at the top or bottom of your device. Looks like a microphone.
Okay.
That's
yeah.
it.
Again, yeah, it's very informative, and thank you very much. The one question I'd like to ask, and it's I know you didn't want personal questions asked, but I did recently get called in 3 weeks ago for a transplant, and having gone to the hospital for maybe 4 or 6 hours, unfortunately, I was then told the kidney was no good. Nobody told me why the kidney wasn't any good, and my nephrologist can't even find out why the kidney wasn't any good.
Oh.
But my question really is, having been called 3 weeks ago, is there a top of this list? In fact, yeah, I'm wondering how long I might have to wait till I get called again.
Okay.
You know, could it be weeks, months, years, or is there a top of the list? Is what I'm asking.
Yeah, unfortunately, no. This is the concept that's quite difficult. It's not a list that you go on and you get to the top and it's your turn. But the way that the national allocation algorithm works, it puts quite a lot of weight behind. It's a point system. And there are 7 factors involved in the point system, and one of them that carries quite a lot of influence is wait time. So you're given a wait time point for every day that you're on the transplant list, and then that is used as part of the allocation. So the longer, if you've accrued quite a lot of wait time, then you get a lot of wait time points. And wait time is calculated from the date that you first go on the transplant waiting list, or the date that you first started dialysis, whichever is sooner. It doesn't necessarily mean that you've reached the top of the list, but anecdotally what tends to happen is when people start getting offers, they do tend to seem to get a couple of offers, and it's probably because of the amount of wait time that they've accrued.
Okay, because you're just thinking, in basic terms, you talk about blood group being sort of, blood group O, which is what most people have got. And the way you've explained how the kidney system is worked out, is it like a blood group? I mean, it was quite complicated the way you were explaining things, but of most people, how can I explain myself? Is it basically like your blood group? If you're group O, you have no problem getting a blood transfusion. So if you've got a certain level of antibodies or whatever in your tissue matching, does it mean that there are more kidneys available than not?
Yeah, so it's sort of the converse in a way. It's if you've got lots of antibodies, it's going to be very difficult for you to find a donor. And if you've got no antibodies, it will be easier to find a donor. Because having an antibody, so say I have an antibody to HLA-A2, that means that I can't have any donor that is HLA-A2. And as I said, that's one of the most common ones. So that would automatically mean that 50% of donors aren't suitable for me. But if I have an antibody to HLA-A80, which is not really found in the donor population very often, I've still only got one antibody, but lots of donors would be suitable for me. It wouldn't exclude lots of donors like A2.
Okay, thank you very much. Thank you.
You're welcome.
Thank you.
And for a final one there, we'll come to Gemma. Gemma, if you'd like to unmute yourself.
Hi, I hope you can hear me.
Yeah,
Yes, hi.
Hello. Thanks, it's been really useful to listen to everything and to get more information, but I suppose this question comes sort of personally. So I've got high antibodies, well, I was told that I have. I've had 2 previous transplants and numerous blood transfusions, but I am currently on the paired exchange list as well as the deceased donor list. Is there ever, and I have been for 8 years, is there ever a point that you get to where they look at like the possibility of using one of your paired exchange donors as a donor direct to me through like
HLA incompatible,
or through plasma exchange, which I did have for my second transplant but didn't work.
Yeah, there used to be a lot more HLA incompatible transplantation going on in the UK, but then the sharing scheme has been so successful that sort of dropped off a little bit in the UK at the moment. But it is still a possibility. It's just it's a bit of almost a bit of playing the numbers, trying to get the best option for you, taking into account how long it might be before you got an offer. If you've reached the point where you've done lots of cycles on the sharing scheme, then you might want to look at a more immunologically riskier option. So the sharing scheme is designed to get you an immunologically safe transplant. If that's not possible through the sharing scheme, then you might start you and your team might start to discuss more above standard risk immunological options.
It's difficult with it being my third. We obviously need the best one possible, but then it's like, do you take a gamble? Or so yeah, I think it's a bit of a complex case. I probably need to raise that question with the team.
I mean, and you know, obviously I don't know your particular status, but then there is this imlifidase enabled transplantation program that's going to start in the UK very soon. But what the imlifidase drug does is just remove all antibodies from circulation so that you can have a transplant in that window of opportunity. But
what's that called again? Sorry.
Imlifidase. I have had to practice saying that.
And are they doing that at Manchester?
It's UK wide. But you've got to bear in mind that this isn't the wonder drug. It's not going to be the answer to everything. It will be a higher immunological risk, which means post-transplant there may be more problems.
Okay, thank you very much. That's been useful. Thank you.
I see we've had more hands raised. Unfortunately, we are out of time for taking any more questions live tonight. Judith, is there anything you'd like to round off with just now?
No, I'm just hope this has been useful for everybody. So I can only talk from the Manchester point of view, but this is stuff that we discuss all the time with the nephrologists and the transplant surgeons. And there will be one of me somewhere in every centre that could maybe help you and discuss your particular case and your particular antibody profile. All my H&I colleagues will probably curse me now.
Please don't mention Judith's name if you're on.
Keep Judith out of it. I mean, actually, can I just sort of jump in? I mean, transplant is, it's a fascinating and actually frustrating subject for those who are awaiting transplant. We will be sending information out at the end of this meeting to the people that registered through the PKD charity, and the NKF will also be sending follow-up information too. And just want to finally say, for any PKD patients online, that in the north, anywhere in the north, we've got a PKD patient day on 11th of November. Please make sure you register for that. It's a free event. We're covering transplant, whole day event, and anybody in the south, in London, southeast, we've got PKD patient day on the 7th of October. Head to our website, go onto social media, you'll find registration links for that there. And again, National Kidney Federation have got a whole host of events coming up. So keep a lookout for there is lots of information out there. Susan, did you want to wrap?
No, I was just going to hand over and see if Pete would like to wrap for the evening. Perhaps he would like to do a little plug for if you're still taking bookings for your patient information weekend.
Yes, so last chance to go to our patient weekend, which is not this weekend, but the weekend after. So if you want to go to our website, kidney.org.uk, watch again for this webinar. If you've got friends or you want to watch it all over again and take it all in, will be on the PKD website and our website, kidney.org.uk. Hopefully, I'll be able to get it on the watch again round about lunchtime tomorrow. So look out for the emails going out. Thank you very much.
Thank you. Good night, all.
Yeah, fantastic. Thanks, everybody. And thanks, Judith.
Thank you.
Thank you.
Good night, all.
Take care.
Have a nice evening. Bye now.